That you all are doing well. Today we are going to look at AS and A level biology, October November 2023, paper 1 2. This is paper 1 and it is of 1 hour 15 minutes. Question number 1 की तरफ move on करते हैं. Let's see that what question number 1 is saying. Which combination of lenses for a light microscope will give the greatest magnification? So magnification basically is the product of the power of lenses. So can we say that magnification is equal to the eyepiece lens value into the objective lens value, right? What we are going to do is that we are going to calculate the value of magnification for each of the option, and then the highest value will be our answer. If we calculate the magnification for part A, it is going to come to be 500. For part B, it is 400. C part in C part, the magnification is coming to be 600, and then finally in D part, it's coming to be 1500. So the greatest magnification is in the option D, where the eyepiece lens is of into 15 and the objective lens is into 100. Hence, our answer over here will be part D. Let's move on. Next question mein they are saying that the diagram shows an electron micrograph of virus particle in a human nucleus. ठीक है, they are showing that it is a human nucleus और ये virus particle है and the magnification of this image is 24,000. What is the diameter of the labeled virus particle? We have been given the magnification in the question and they are asking us to calculate the diameter of the labeled virus particle. So can we apply the formula K magnification is equal to image length divided by the actual length? The magnification is given to us as 24,000. How are we going to calculate the image length? Simply we are going to take a scale and we are going to measure the image length. They are asking us to calculate the diameter. So diameter के लिए we are going to measure this length of the virus. Whatever the answer is going to come, you are going to write it over here. And always remember, when you are using anything in the calculation while calculating the magnification, you are going to use the length in the micrometers. ठीक है? And then whatever is the actual length coming will be your answer. The answer in the marking scheme is coming to be D, which is 1.5 into 10 raised to the power 2 nanometers. ठीक है? You guys just have to solve it on the paper and then you are inshallah going to get the same answer. Question number 3 is stating that what is correct about the synthesis and release of glycoprotein mucin in the goblet cell. We all know that what is glycoprotein. Glycoprotein is glycogen plus protein, right? Statement 1 is saying that the protein is produced on ribosomes and the carbohydrate chain is added in the Golgi body. जी बिल्कुल, the protein is produced in the ribosome. This is correct. Golgi body, we all know this is used for the packaging, modification, collecting of the molecules, right? So when the carbohydrate chain is added to the protein, so this is basically the modification, right? Modification हो रही है protein की. So option 1 is correct. Statement 2 is saying the glycoproteins are packed into vesicles in the Golgi body forming lysosomes. No, this cannot happen. Why? Because lysosomes, what are the lysosomes basically? Basically, the lysosomes are the suicidal bags. Suicidal bags, they contain the hydrolytic enzymes H2O2. What do basically the lysosomes do is that they do intracellular killing of the cell organelles that which are worn out or which are in excess. So this cannot be the answer. Why? Because the glycoproteins are not packed into the vesicles in the Golgi body forming the lysosomes. Statement 3 is saying that secretory vesicles containing glycoprotein move from Golgi body and fuse with the cell surface membrane. Yes, this is correct. Why? Because, for example, if we say that this is the Golgi body, this is the cis phase and this is the trans phase. What happens over here that vesicles come over here and then they fuse with this membrane And at the trans phase, what happens that the vesicles bud off and then go to the cell surface membrane and fuse with them. So the answer over here is 1 and 3 only. Option B is our correct answer. Question number 4 says that what are the functions of the microtubule? So basically, what are the uses of the microtubule? That microtubules form the cytoskeleton. Microtubules form the cytoskeleton and allows the movement of the organelles. They're also used to make the spindle fibers, right, in the cell division. Movement of organelles. Now let's see that which statement is complementary with the uses. Allowing movement of cilia in the bronchus. Bilko statement 1 is correct. Allowing the movement of the cilia in the bronchus is done because of the microtubules, because microtubules allow the movement as well. Attaching to centromeres during metaphase. What happens in the metaphase that spindle fibers go and then they attach to the center of the chromosomes, right? So this option is also correct. Moving secretory vesicles around the cell. Yes, movement of the organelle is a function of the cytoskeleton. Sorry, the function of the spindle microtubules. So this is also the right answer. Hence the answer over here is coming to be A. All the statements are correct, 1, 2 and 3. Question number 5 is saying that which organelles contain nucleic acid. The options here are given as chloroplast, mitochondrion and Golgi body. We all know that what are basically the Golgi bodies? They are the stacks of curved flattened sacs, right? Like this. They do not contain the nucleic acid. Golgi body do not contain the nucleic acid. Mitochondrion and chloroplast, we all know that they have the circular DNA within them. The location for the circular DNA in the mitochondrion is the matrix, and the location for the circular DNA in the chloroplast is the stroma. Hence, the nucleic acids are only found in the mitochondrion and the chloroplast. The answer that we will be getting over here is D. Number 6 says that some features of the cells are listed. Cell wall, cell surface membrane, ribosomes. Which features can be found in plant cells and in the prokaryotic cells? Now they are saying that what are the common features of the plant cell and in the prokaryotic cells. We all know that the prokaryotic cells are the bacterial cells, right? They are the bacterial cells. Now they are saying that what is common in the plant cell and the prokaryotic cell. Yes, we all know that cell wall is common in both the plant cell and the prokaryotic cell. अब जो plant cell की cell wall है, it is made up of cellulose and prokaryotic cell wall is made up of peptidoglycan. Cell surface membrane is found in both the cells. Ribosomes. Yes, are ribosomes found in both the plant cells and the prokaryotic cells? जी, बिल्कुल, the ribosomes are found in the plant cell as well as the prokaryotic cell. Plant cells have both the 70S and 80S ribosomes, and the prokaryotic cells are having only 70S ribosomes. So hence the answer over here is coming to be part A, in which 1, 2 and 3, all three statements are correct. Question number 7 says that which bonds are present in all viruses. We all know that the basic structure of the virus consists of a protein coat, which is also known as capsid, or the nucleic acid, is either DNA or RNA. And also in some of the viruses, there is an envelope and spikes found. Bonds ki, they are asking about the bonds in this question. So statement 1 is saying phosphodiester bonds. We all know that the nucleic acid contains the phosphodiester bonds, peptide bond and the covalent bond. Peptide bonds are found in this capsid. It contains the protein, right? It is made up of protein. So it contains the peptide bonds as well. We all know that the phosphodiester bond and the covalent bond, they are both phosphodiester bond and the peptide bonds are both the covalent bonds. So hence over here the answer is going to be A. Question number 8 says that a student carried out the Benedict's test on the four different concentration of glucose solution and then recorded the time taken for the first appearance of a color change, the endpoint. The student found it difficult to identify the first appearance of the color change and consistently timed each condition for 2 seconds after the color change first appeared. This introduced a source of error into the experiment. So basically what the student is doing that he is timing the solution after 2 seconds of the color change, which should not be done. What should be done is that whenever the first color change is appeared, he should stop the stopwatch. So basically this error which is introduced over here is known as the systematic error, which is the error introduced due to the fault in the method of experiment. Which statement about this error are correct? The effect of the error will be reduced if the student performs 3 repeats at each concentration of glucose. This is basically a systematic error. So systematic errors cannot be resolved by repeat and average. So statement 1 is incorrect. The error will prevent the student from identifying which solution has the highest concentration of glucose. No, even if he time it after 2 seconds, he is going to identify that which solution has the highest concentration of glucose. So this is also incorrect. Statement 3 says that the error is systematic as the student consistently timed each solution for 2 seconds after the endpoint. Yes, this statement is correct. Why? Because he should not have timed it after 2 seconds of the endpoint. Instead, what he should do is that he should immediately stop the stopwatch when the first color change appears. He should not wait for further 2 seconds. Hence, the answer over here is option D, 3 only. Question number 9 says that which statement about amylopectin and glycogen are correct? If I talk about them, they are basically the polymers of alpha glucose, right? If I draw the structure of amylopectin and glycogen, you can clearly see that glycogen is more branched than the amylopectin. At the point where the branches occurred, it is having alpha 1,6 glycosidic bond, and over here they are having the alpha 1,4 glycosidic bond, right? Statement 1 is saying amylopectin and glycogen contains alpha 1,4 glycosidic bond. जी बिल्कुल, both the amylopectin and glycogen consist of alpha 1,4 glycosidic bond. Amylopectin contains beta glucose. No, this is not the case. Why? Because they are the polymers of alpha glucose. Statement 3 says that glycogen contains more 1,6 branches than amylopectin. You can clearly see that glycogen is more branched than the amylopectin. So statement 3 is correct. So answer over here is going to be option B. 1 and 3 are the correct statements. Question number 10 says that how many statements about fatty acids are correct? Statement 1 is saying fatty acids are either saturated or unsaturated. Yes, this statement is correct. They can be saturated or unsaturated. Fatty acids have at least two double bonds. No, this statement is incorrect. Why? Because fatty acids can be both monounsaturated and as well as they can be polyunsaturated. Polyunsaturated means that having more than one double bond, and monounsaturated means having only one carbon-carbon double bond. Statement 3 says that long chains of fatty acids are very good stores of energy. Yes, this statement is correct. Why? Because the long chain fatty acids have more carbon-carbon bonds and hence they can store more amount of energy than the shorter chain fatty acids. So statement 3 is also correct. Statement 4 says that fatty acids are insoluble in water and do not affect the water potential of the cell. We all know that fats are insoluble in water, and anything that is insoluble in water does not affect the water potential of the cell. Hence, our answer over here is option C. Three statements are correct. Question number 11 says that the diagram represents an amino acid. R represents a variable side chain. What is not a possible side chain? You can clearly see in option D, which structure I have drawn over here, that all the carbon atoms in the side chain of D have four covalent bonds already, so it can't be bonded as a side chain to the amino acid. If you look at option A, B, and C, there are at least one carbon atom in the chain with three covalent bonds. What does this do is that this allows one bond to be formed to the amino acid as the side chain. Hence, the answer over here is part D, as there is no free covalent bond in any of the carbon atom over here. Question number 12 says that the graph shows the effect of an enzyme on a reaction. So this is basically the energy time graph. They are saying that which row identifies X, Y, and Z. We all know whenever we use an enzyme or catalyst in a reaction, the activation energy decreases. So the graph of the Y is showing a decreased activation energy. So we can say that Y is a catalyzed reaction. X is the uncatalyzed reaction. Why? Because it is showing a high activation energy. Z is basically the delta H, which is the overall energy change during the reaction. So X represents the uncatalyzed reaction, Y is the catalyzed reaction, Z is the overall energy released during the reaction. Hence the answer over here is option D. Question 13 says that the graph shows the results of two experiments on the effect of increasing the substrate concentration on the rate of an enzyme catalyzed reaction. One experiment was at a high concentration of enzyme and the second was at a low concentration of enzyme. All the other variables were standardized. So they are showing the graph of the rate of reaction versus the substrate concentration. What would limit the initial rate of reaction at point X? So they are basically asking that what is limiting the rate of reaction at point X. We can clearly see that as we are increasing the concentration of enzyme, the rate of reaction is also increasing and then it is becoming constant again. So we can clearly say that enzyme concentration is limiting the rate of reaction. Why? Because even though there are a lot of substrate present, if the enzymes are not present, then the enzyme catalyzed reaction will not take place. Hence, the answer over here is option A. Question number 14 is saying that which statement about the Mitchell's maintain constant Km of an enzyme are correct. If I draw a graph from which we can deduce the Mitchell's maintain constant Km, we know that the graph is between the rate of reaction and the substrate concentration, right? The maximum rate of reaction is termed as Vmax, and half Vmax is basically the Km. So we can say that Km is the substrate concentration at half Vmax. What happens at the Vmax अगर हम बात करें, so at Vmax the enzymes are working at their maximum possible rate, which means that all the active sites of the enzymes are filled with the substrate, and at half Vmax 50% of the active sites of the enzymes are filled with the substrate. क्योंकि because at Vmax 100% of the active sites are filled with the substrate, at half Vmax 50% of the active site would be filled with the substrate. So statement 1 क्या कह रहा है? At the Km value, half the active sites of the enzyme should be occupied by the substrate. Yes, we have discussed it. Statement 2 is saying Km represents the substrate concentration at which the enzyme is working at half its maximum rate. Yes, this is also the point we have discussed above. Now, in statement 3, they are saying the lower the Km value, the lower the affinity of the enzyme to its substrate. अब यहाँ पे what point we should understand over here is that whenever there is the lower Km value, when there is the lower Km value, it means that lower the substrate concentration is required to fill the 50% of the एक्टिव साइट्स ऑफ द एंजाइम विद द सबस्ट्रेट, तो हमारे पास एफिनिटी क्या हो जाएगी, हायर, higher affinity between the enzymes and the substrate. And alternatively, when there is higher Km means that more substrate concentration is required to fill the 50% active sites of the enzyme with the substrate. So, the affinity between the active site of the enzyme and the substrate is lower, right? So higher Km में lower affinity. So hence option 3 is incorrect. Option 4, statement 4 देखते हैं that when an enzyme has a high Km value, the enzyme catalyzed reaction will proceed very slowly to its maximum rate. जी बिल्कुल, this is correct. Why? Because at higher Km value, there is lower affinity between the enzymes and the substrate, and when there is lower affinity between the enzymes and the substrate, then the reaction will proceed very slowly to its maximum rate. So the answer over here is option B. 1, 2, and 4 statements are correct. Question number 15 says that which component of the cell surface membrane helps to reduce the fluidity of the phospholipids at higher temperature? We know that whenever we are having a higher temperature, so the fluidity of the cell surface membrane increases, right? And now they are asking that we have to reduce the fluidity of the cell surface membrane. Phosphate groups have no link with the fluidity. Proteins have no link with the fluidity. Unsaturated fatty acids. We know that the presence of unsaturated fatty acids increase the fluidity of a cell surface membrane. So it will not reduce the fluidity or else it would be increase in the fluidity. Why? Because whenever there are unsaturated fatty acids, so they introduce a kink, right? And when it introduces a kink, it increases the fluidity. So our answer over here will be option C, which is cholesterol. Why our answer over here would be cholesterol? Because the cholesterol prevents the membrane from becoming too fluid or too rigid. We can say that it controls the fluidity of the membrane. Question number 16 says that plant cells were left for 15 minutes in three different sugar solutions, 10%, 5%, and 1%. The water potentials in the cytoplasm of the three cells was the same as at the start of the experiment. The diagram shows the appearance of the cells after 50 minutes using a light microscope. This is cell P, this is cell Q, and this is cell R. We can clearly see over here that cell Q is in a turgid state. Okay, cell R is in a plasmolized state. What is basically the difference between plasmolized and the flaccid cell? The basic difference between is that in plasmolized, the cell surface membrane is basically detached from the cell wall, and in flaccid, the cell surface membrane is not detached from the cell wall. Cell P is flaccid. Which conclusion is correct? Cell P has the same concentration of sugars inside and outside the cell. No, this is not the case, as they are saying that 10%, 5%, and 1% sugar solutions are used. Cell Q is flaccid and cell P is plasmolized. No, cell P is flaccid and cell Q is turgid. The sugar solution outside the cell R has a less negative water potential than inside cell R. We know कि cell R में the water is coming out of the cell. It means that inside the cell there must be a higher water potential and outside the cell there must be a lower water potential. If there is a higher water potential inside the cell, it means that the water potential inside the cell is less negative, and if there is a lower water potential outside the cell, it means that it is more negative. ठीक है? So the answer over here is option C, that cell Q was placed in the 1% sugar solution. If it was placed in 1% sugar solution, it means that it is having higher water potential outside the cell, and then the water has been taken up by cell Q. Question number 17 is saying the graph shows the effect of increasing the side lengths of agar cube on the surface area and the volumes of the cube. If we talk about the surface area of the cube, तो surface area of the cube हम लोग किस तरह से find out करते हैं? 6 लेंथ या फिर साइड का स्क्वायर, और वॉल्यूम किस तरह से फाइंड आउट करते हैं? लेंथ या फिर साइड का क्यूब. Now they are saying that which row correctly identifies line 1, line 2 and the effect of increasing the side lengths on the surface area to volume ratio of the cube. If we know कि अगर if we take some value के let's assume the side of the agar cube before it increases to be 5, ठीक है? We are assuming it to be 5. If we take 5 over here, तो हमारे पास surface area, the value of the surface area which we will get is 150 and the value for the volume which we will get is 125. And then let's assume that the side increases to 10, right? And when the side increases to 10, we got a surface area to be about 600 and we get the volume to be about 1000. These are the assumptions we have made. So what we can see over here, that when we increase the side length from 5 to 10, then the surface area have become less than the volume. So can we say that as we are increasing the side length, the surface area is becoming less than the volume. So this line, which is line 1, may be representing the volume. Can we say this? Why? Because as we are increasing the side length, we know that volume is increasing and surface area is comparatively decreasing. So we can say that line 1 is the volume and line 2 is the surface area. If we talk about the surface area to volume ratio before the side length of the cube increases, we know that surface area to volume ratio can be calculated by dividing surface area value with the volume ratio value, and if we do it we get an answer which is 1.2, right? And then if we calculate the surface area to volume ratio of the cube when the side length is increased, we get the answer to be about 0.6. So we know that the surface area to volume ratio has decreased when the side length is increased. So hence the answer over here would be option C. So question number 18 over here says that a human embryo consists of 12 cells at the start of day 1. The cell divides by mitosis for 4 complete days at a rate of 1 division every 32 hours. What is the maximum number of cells in the embryo at the end of day 4? So what we over here know is that for one division to take place, it requires 32 hours. So what will be the number of divisions that we are going to get over 4 days? In 4 days, how many hours are there? If we talk about the number of hours in 4 days, it is 96 hours, right? So in 96 hours, how many number of divisions will happen? We are simply going to cross multiply it and we are going to get 3 divisions will happen over a tenure of 4 days, right? 3 divisions will happen, which means that if we talk about mitosis, then mitosis is a type of a nuclear division in which a parent nuclei divides to form two identical daughter nuclei, which are identical to each other as well as the parent nuclei. Then further, this cell divides into two more cells, which is known as the second division. This was the first division, right? Then this was the second division, we can say. And then further, this individual cell is going to divide into two more identical cells, which we can say is the third division. So how can we solve this question that at the start we were having 12 cells, right? So simply after the first division, we are going to have 12 into 2 cells, which will be 24 cells. Now these 24 cells are going to again divide into one more cell, which we can say will be 48 cells. And then this one cell out of those 48 cells is again going to divide into two cells. So we are simply going to multiply 48 by 2. I hope that you guys are getting how to solve it. We simply have to multiply the values with 2. So hence the answer over here is option B. 96 cells will remain. Question number 19 says that which diagram shows the role of mitosis. If we talk about the role of mitosis in general, we know that the role of mitosis is growth, repair and replacement of the worn out cells. In part 1, they are showing us that genetically identical offsprings are there. We know that mitosis produces genetically identical offsprings. So we can say that part 1 is showing the role of mitosis. In option 2, what is happening that a bud is simply opening. So over here we can also say that this is also mitosis. Why? Because growth is being shown. Option 3 is part 3 is also the growth. Why? Because it is increasing in size. Part 4 can be, we can say that part 4 is the replacement of the worn out cells. So hence the answer over here is option A, 1, 2, 3 and 4. All parts are showing the role of mitosis. Question number 20 says that which over here says that which events are a part of mitotic cell cycle. If I completely draw the cell cycle over here, we know that in cell cycle there is a G1 phase, there is a S1 phase, there is a G2 phase. This I am drawing a very rough cell cycle. This is the M phase over here. What comes under the M phase? M phase is also known as the mitosis. About that which phases come under mitosis, then it must be prophase, metaphase, anaphase, and telophase. Over here comes the cytokinesis. Now they are saying that which events are the part of mitotic cell cycle. We know that interphase is a part of the mitotic cell cycle. What is interphase over here? From G1 to G2, we can say that it is a interphase part of the mitotic cell cycle. Anaphase is also a part of mitotic cell cycle. Cytokinesis is also a part of mitotic cell cycle. Yes, interphase and cytokinesis are not a part of mitosis over here, but they are the part of the mitotic cell cycle. Hence, the answer over here is option A. 1, 2, and 3 are correct.